Hello, my name is Benjamin Bravo, and I will talk to you today about wildlife cameras in ecology. This is the second part of a video series that I am making on wildlife cameras, and the purpose of this video is to use the scientific method to answer questions, ecology questions, using camera trap data. The camera trap data used for this video is from the Arastradero Preserve camera trap project, and this video is meant for high school students. It can be about an hour-long lesson if you want to expand from this 15-minute video. In order to do this, you should have a basic understanding of camera traps and the scientific method. So I am a recent graduate of the Stanford Earth Systems program, master's program, with an emphasis in conservation biology. I used camera traps for my own research on the dusky-footed wood rat, as well as my research on deer and coyote. And I was part of a photo tagging volunteer group at Stanford's Jasper Ridge. This is me shown in the right here. This photo was taken by a camera trap. And so what we'll be talking about today is actually somewhat similar to what I used to the scientific method that I use for conducting my own research with camera traps. So the scientific method can be broken down into a few very simple steps. This is a slight modification of what you may have seen in high school, but this is very similar to how papers are laid out in scientific research. So first we will start with the introduction. So in the introduction you want to decide on a general topic, a location as well, and you want to also do some background reading on the basic topic. You might want to read other papers that have done similar projects to the project that you would like to do, and you definitely want to look up information on animal guides on various websites. If you are using data derived from the Arastradero Preserve camera study, it's good to know a bit about the preserve. So Arastradero Preserve is located near Palo Alto. There are quite a few preserves near Arastradero Preserve which could be sources of animal populations, and Arastradero Preserve is also located in a fairly high density human area, and it has quite a bit of human usage on the various trails. These are some of the species that are found at the preserve, and if you would like to do a study on a specific species, you can pick one from the list shown here. It's good before you start a study to have done a pilot study or to have information from other studies that have been done in the same area on similar species. So for example, if you are doing your own study with camera traps at Arastradero Preserve, you should know about what has already been found. This chart just shows the number of events of different animal species found at the 5 different sites where the cameras were located. So this just shows you that you can't do a study on any species. You might want to pick a species that is present at many of the sites. So for example, skunk would be a difficult animal to study because it's only present at maybe 2 or 3 of the sites. So you might want to pick an animal like deer or coyote, which are present at pretty much all the sites. So once you have a general question in mind, you should fill out the section 1 on the worksheet, and now we'll move on to the hypothesis, which is section 2. In order to form a hypothesis, you must have a dependent and independent variable already selected. A dependent variable is the thing that you will be measuring, and an independent variable is the variable that causes change in the dependent variable. So we can see the dependent and independent variable in the research question. So the research question is what you want to understand, and so in the research question we say what effect do humans have on coyote activity, humans being the independent variable and coyote being the dependent variable. In your hypothesis, you predict the answer you expect to your own question. So if I were studying coyotes and humans, I would predict that there will be a lower coyote activity in locations where there is a higher human activity. That's my sample hypothesis. And then you also want to describe why your research is important, which knowledge gap you think it can fill, and possibly how it can apply to conservation. Next, we move on to methods. So for the Arastradero study, we used camera traps. Camera traps are a great observational tool to use in ecology. They can tell you when animals are present, where animals are present, which species are likely to be present at these sites, and they can give you a rough approximation of how many animals are present. This is very rough and is more useful to tell you if the number of species is changing over time, or if the number of individuals of one species is approximately changing over time. And so these are our methods that we used for the pilot study at Arastradero Preserve, and these are where the cameras were located. As you can see, they're kind of clustered in the center of the preserve. So with an ecology study using camera traps, the actual experiment is the deployment of the cameras. You are leaving the cameras in place for a certain amount of time, and that is when they are collecting data. During this time, you do not want to disturb the cameras, and then after you are done with the sampling period, you want to take the chips from the cameras, put them into a computer, and download the photos. You can also collect more information about the specific sites where the cameras are located. Usually this is environmental information such as like canopy cover, distance to trails, or distance to humans possibly, closeness to the edge of the preserve, closeness to water. You can also look at specific type of plant species that are in one location. And a lot of these can be used for describing habitat preferences by the animals that show up in the camera trap photos. Okay, next step is analysis. So by now you should have filled out the whole front page of the worksheet if you are following along. Analysis is really the main section. So for analysis, the first step is to actually label the photos. This can be done using a website such as Zooniverse, which I talked about in the first video, or you can also use another website like WildID or just an Excel sheet. So what you need to do is label all the photos. Zooniverse allows you to classify the photos from a list of species, and it's very user-friendly, and I recommend it. From Zooniverse, or once you're done classifying the photos, you'll get a sheet that has the date the photo was taken, the time, the species. It'll also have a unique ID of some kind, the name of the sampling event, in case you have multiple time periods when you are sampling the photos, as well as the site, or in this case I called it the camera. Now you can't usually use this information itself to start analyzing your data. You have to do a little bit of processing. And so processing is just kind of consolidating and summarizing all those lines of data into something that's a little bit more usable. So you can do this with computer code, you can do this with Excel sheets, pivot tables, and what you want to end up with is something a little bit more like this. Now this is very similar to the sheet that I have attached to this video, a sample data sheet, and this shows the camera number or the site, the number of events of the subject species. It also shows number of events of another related species you might be analyzing. It shows maybe some habitat variables. And I should clarify that events here refer to the number of visitations to the camera by one specific animal. Events are a better unit to use than raw number of photos, because an animal can visit a site many times in a row during a short time period, but that really only counts as one visit to the site. And this is a good, events are a good unit to use in camera trap studies for describing the relative abundance. Also note in this table, probable independent variables are shown in red, and the dependent in this situation is the coyote events. This is because I am talking about coyotes as an example for this video. So from that data you can create a scatter plot, and from that scatter plot you can do a linear correlation. This is a very simple statistic test, or plot and a test. And so I have my independent variable on the bottom, which are number of human events, and my dependent variable on the y-axis, which are a number of coyote events. So I'm trying to see if you increase the number of human events, or you get an increase in the number of coyote events. This is to kind of see if coyotes may be avoiding sites where humans are located. Each point on this plot represents a specific camera. So since we had 5 cameras, we only have 5 data points for this plot. So we can see they are positively correlated. This correlation is not really that significant, but it will allow us to maybe hazard an answer. You can also do a more advanced test in R and create something called an overlap plot. This just shows the times when the species are active. And so we have humans shown in red, coyotes shown in black, and you can see where they overlap in the times they visit sites. So it's possible there's an interaction between humans and coyotes. Another plot you can make that's very useful, this would be answering a different question than what I was talking about for most of the presentation. This is trying to see what the possible effect the habitat type could have on the coyote visitations, or coyote relative abundance. This could be used for trying to describe a possible preference the coyote may have for one type of habitat, for sites located in one type of habitat. On the left we have a simple dot plot, and on the right we have a box plot which shows things like percentiles and the median. And next, so now I recommend you stop the video and fill out the graphs, fill out the tables, and then once you are done, we can move on to the next step. I also have interactive plots, which can be very useful for playing around with the data. So the last step is the conclusion. In the conclusion, you say what you found. You can say if your stats were significant, and you can also use that to say if your hypothesis was correct. This is also a good time to give a possible explanation for the outcome. And in order to really give a good explanation, you want to do some more reading in the literature, read some more sources, try to see if other studies have found something similar and why they may have found something similar, or if other studies have found something different. It's also really good at this point to say future studies should, and then describe what future studies could do if you had more time, more resources, or if you want to answer a slightly different question. So I have an example shown here of what that could look like. So that's pretty much all I have for this lesson. So hopefully by now you have filled out most of the worksheet. And if you want to get more out of this lesson, I recommend you spend more time discussing each of the sections. You can do more statistics tests. You can ask multiple questions of the same topic. You can also do some really in-depth discussions of the effects the cameras may have on the animals, and if camera traps are actually a good tool to use. I'm a little bit biased. I love camera traps. I think they're a great tool to use, but there are some limitations that could be considered. You can also collect more data. You can set up your own camera traps if you want, and collect your own original data in a different location. And you should also brainstorm some possible future studies you could do if, say, you were at a university and maybe you had a few years to do a longer term study. I just want to throw out some brief disclaimers. I only used 5 cameras for the study, so the data collected from this study is not quite usable for more advanced, more significant statistics. And the lesson plan that I designed here, while it is pretty useful, there are other lessons plans which are cater more towards specific scientific standards. Some of those lesson plans I have linked here, which may be based off some state standards. And so I would like to thank you. If you are part of the camera trap project at Arastradero Preserve, I highly encourage you to label more photos, and I invite you to consider doing your own camera trap study at Arastradero Preserve if you talk to Avi and you talk to someone there. So thank you. Have a good day. Bye.